Alright, now we are on to transcription. Part 3, we talked about how DNA replicates itself. That's important. Occurs during the S phase, interphase, and is important so that when new cells when cells divide, that each daughter cell has a complete set of DNA. Today we're going to talk about the first step in getting DNA, basically how we go from DNA to genes, or different traits that we have. Alright, so learning target 8, compare and contrast DNA and RNA. Understand transcription and the role that RNA polymerase plays in it. And then know how eukaryotic RNAs are modified before leaving the nucleus. Okay, so maybe you're thinking, why in the heck are we talking about RNA when we're talking, this whole unit is, DNA. All we've talked about so far is DNA. DNA history, DNA structure, DNA replication. What does RNA have to do with it? Actually, RNA has a lot to do with DNA. Okay, just first some differences. RNA is single-stranded, whereas DNA is double. RNA is found in the nucleus and the cytoplasm in both places. DNA is only found in the nucleus, at least in eukaryotic organisms. Okay, DNA is similar to RNA in that both are composed of nucleotides. Each nucleotide has 3 parts, just like the nucleotides in DNA. Okay, RNA has a phosphate group. It's got a 5-carbon sugar, only this sugar is different. It is ribose. That is why it's RNA, for ribonucleic acid. So RNA. Alright, there are 4 possible nitrogen bases: adenine, guanine, cytosine, and uracil. Okay, so uracil is the new thing. It is what replaces thymine. There is no thymine in RNA. Alright, gene expression. A segment of DNA that is going to code for a polypeptide. And if you think back to our organic chemistry unit, polypeptide, what it is it's a bunch of amino acids. The bonds that are between those amino acids are called peptide bonds. Poly meaning many. So you've got this nice string here of, it's going to do all sorts of crazy things depending on its shape. And that's a whole bunch of jumble. Okay. So there are different types of RNA that we need to talk about, and then we'll talk a little more in depth about them as we go through transcription and then later translation. So there's mRNA. The little m stands for messenger RNA. This carries genetic information from the DNA out to the ribosomes, because remember, the DNA is found only in the nucleus. Our messenger RNA is very appropriately named because it's going to be carrying the genetic message. Okay, a genetic copy, only an RNA outside of the nucleus. Okay, RNA, or rRNA, this is what ribosomes are made out of, and it is where proteins are being assembled. And then there's tRNA. tRNA, they bring in amino acids to the ribosome. So if you look here, this is going to be mRNA here, this little strand here. rRNA is going to be the 2 parts of the ribosome. And then tRNA, that's this funny looking thing here that actually almost looks like a T. Okay, so let's talk about transcription. Transcription, this makes an RNA copy from the DNA template. There are 3 phases: initiation, elongation, termination. Okay, so if you take a look at this picture over here, it says transcription is taking place here. mRNA nucleotides are being joined by the enzyme RNA polymerase. Okay, so RNA polymerase is this stringy thing back here. Okay, that is combining the nucleotides together. Being joined by the enzyme RNA polymerase so that their sequence is a complementary strand of DNA, or complementary strand to that of the DNA. This mRNA transcript is ready to be processed, and then it will move into the cytoplasm. So this light blue thing here, it's single strand, and this is what's going to exit our nucleus out into the cytoplasm. Okay, so RNA polymerase binds to a promoter region. Okay, so this right here, this would be a promoter region. It's where the RNA polymerase will bind to, because it's what's going to promote or going to start this whole transcription process. DNA helix unzips because the RNA polymerase, it unzips. The RNA polymerase is then going to add complementary RNA nucleotides in. So for T, we're going to have A. T, we're going to add an A. For C, we're going to add G. Okay, and take a note here. A, we're going to replace it with a U, because remember in RNA there is no T. Okay, so we need to talk about processing of this mRNA. Okay, mRNA, it's going, well, first of all, we're going to start here with the DNA. This is the DNA that's going to be transcribed. This is the section that holds the genetic information that we want to be coded for. Okay, now there are these things called, let's see, yes, there are these things called introns and exons. Introns you can think of as basically garbage. They're left over from evolutionary history, bits of DNA or random stuff that are random sequences of genes that we don't need. Okay, and our DNA is actually full of quite a lot of this. It is thought that the introns are basically leftover genes that we no longer need, but somewhere far, 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 far away in the past, our ancestors may have coded for something that they needed. Okay, so I always remember this as introns. They go in the garbage can. It's garbage. Body doesn't need it. Okay, and exons. Okay, exons here, see E. Exons are what are going to exit. Okay, exons exit the nucleus. Okay, so the intron sequences are removed. Ribozyme, this is an RNA enzyme. It's going to splice, or is going to cut the exon segments. The intron segments out and splice those two exon, or 1, 2, 3, those 3 exon segments together. Okay, so you've got your primary mRNA that has exons and introns. And then this ribozyme is going to come in and it's going to cut out the introns. Going to throw those in the garbage. Boop, bye-bye. Okay, throw it in the garbage, and then it's going to put all those exons together. Okay, it's very nice. It's a way to condense things so that when the ribosomes are going through and adding amino acids, they're not adding in useless amino acids. Okay, they're getting the exact what is needed. Okay, so that's transcription. Pretty much transcription, you go from nucleus to the cytoplasm. You have your DNA in the nucleus. That's going to get copied into primary mRNA. Ribozyme. Wow, I really can't write, sorry. Ribozyme is going to cut out introns, throw them in the garbage, and then you're going to have your mature mRNA that has only exons, and your exons are what are going to exit the nucleus. Okay, I guess I should add the ribozyme is cutting out introns and putting exons together. Okay, alright, that's it. Okay, so I actually thought of something else that I want to share with you, because you're going to need to be able to do this. Okay, you're going to need to be able to go from DNA to mRNA. This process of DNA to mRNA, and here I haven't left myself enough room, so sorry, this is called transcription. Okay, so let's say I have here a DNA strand A, G, C, T, 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 A, T, C, C, G, A? I don't know. Okay, we're going to talk about this a little bit later, but there are these things called codons. Codons is simply a set of 3, so look, I need G, C. Okay, a set of 3 nitrogen bases. So you're going to need to be able to transcribe the DNA into mRNA. So in order to do that, okay, remember RNA does not have a T, so A's are going to be paired with U's, G's with C's. Okay, C's, look, there's my first codon, G, A, my second codon, A, U, A, G, G, C, U, C, G. Okay, now that is then going to take us to our next step, which is the next section of notes, which is translation. Not too hard. All you have to be able to do is match the A's with the U's and the G's with the C's. And that will get you transcription. Okay.